So in essence, what I'd like to do in beginning today is to revisit what Susan talked to us a bit about, which is the mission of the Data Monitoring Committee. And I've always thought that this example, this experience that we had from the Committee of Program and Clinical Research in AIDS from the 007 new combo trial was a good example of that. And you'll see in my presentations today a lot of examples from the experience of the Data Monitoring Committee for NIAID sponsored trials conducted by the AIDS Clinical Trials Group, or in this case, the Committee Program for Clinical Research in AIDS. I had the privilege of serving on that committee for 20 years, from 86 to 06. We monitored at least 50 trials during that time. Dave DeMetz was on that committee with me. Susan was actually at Division of AIDS during a lot of that time period. So in essence, this is a trial that was a really important stepping stone in HIV. We had to walk before we could run. We first got zidovudine or AZT. Then we moved to two-agent combination, and that's what this study was looking at. The middle group was looking at zidovudine alone, and we were looking at what happens when you compare AZT or zidovudine alone with the addition of DDI or with the addition of DDC. It was a great design in the sense that we could use the common control for both of these interventions. And yet the problem was we wanted to blind it. And so the DDC patients had to get a DDI placebo, the DDI had to get a DDC placebo, and the folks in the middle had to get DDI and DDC placebos, which was going to be something like 18 or 19 placebo pills a day. So Jim Neaton had a clever idea, and that was, we'll take these 1,200 patients, 400 against 400 against 400, and we'll do an unblinded randomization up top. So half of you will know, 600 of you will know you're on the DDI side, and 600 of you will know you're on the DDC side. But everyone within the DDI side in a blinded way would have a 2-to-1 randomization to active DDI against DDI placebo. And everyone on the DDC side would have a blinded 2-to-1 randomization to active DDC against DDC placebo. And then we would pool the placebo arms and compare the 400 getting combination DDI against the placebo and combination DDC against placebo. And the lovely approach with this is we cut the number of placebo pills by a factor of 4. So that was the design. And the study was conducted from 1992 to 95. And we were about a little more than a year into the trial. We were meeting every 3 months, and we met in August of 93. And at that meeting for groups A and B, there were twice as many progression death events, where these were symptomatic AIDS events, Kaposi sarcoma or opportunistic infections or deaths. And deaths alone were 4-fold more. Yet our data monitoring committee looked at this and cautiously said, okay, but let's go further. This is an interim analysis. It's not fully reliable. But when we met 3 months later, that difference in progression to AIDS death events persisted, although not quite as striking as before. But the deaths were even more striking, 17 to 2. And when you looked at all events, meaning each person could have multiple events when they would have repeated events of Kaposi sarcoma or opportunistic infections or deaths, there were twice as many events. And so what should we do? And if we had a lot of time, I would say, what would be your questions that you would ask right now? And one question that I hope you would ask, based on discussions from Susan and Janet yesterday and the day before, are, well, the data monitoring committee needs to know what A and B are. Well, okay, but let's surely hope that when you tell me what A and B are, that this worst group is going to be zidovudine alone, and the better group will be when you add DDI and DDC. But you know, that can't be true, because the DDI and DDC groups, when you pool them, are twice as large as the zidovudine alone. So when you unblind yourself, interestingly, groups A and B are the 2 placebo groups. The 2 placebo groups. There had been no intention when this study was designed to compare DDI placebo to DDC placebo. This was simply an artifact of the approach of basically saying, we're going to have a pooled control group to assess both active DDI and active DDC. And so in essence, the issue then became, what do we do as a data monitoring committee? And I do want to, in fact, invite all of you to serve on this committee with us here today. I'm going to circle back to this later on today in the presentation. We have time to ask your opinion of what we should do. And the reason this is really perplexing is, how can you continue to randomize 2/6 to DDI active, 1/6 to DDI placebo, 1/6 to DDC placebo, and 2/6 to DDC active when you have such evidence of adverse results in the DDI placebo? And the DDI placebo has no upside to it. It's simply a placebo that's being used to create the blind. And so from an individual perspective, safeguarding patient interests, how do we not take action here? And yet, from a perspective of preserving trial integrity and not over-interpreting the results, we have to realize that if you argue that this DDI placebo group should be ignored or can't be used as a control because DDI is potentially harmful, then instead of doing the planned analysis of saying, okay, DDI active has 55 events against 70 control events, you're going to compare to the DDC placebo alone, in which case you're not getting any benefit, even with the 2-to-1 randomization. It's an exact equality of event rates. And instead of being equal on death, 18 against 19, you're going to have the impression that you're harming on mortality. So the interpretability of this trial, if we prejudge these results, could be compromised. And by the way, there were many other trials going on that didn't have 2 combinations against zidovudine alone. It was just looking at DDI combination against zidovudine alone in this manner. Those trials had no way of assessing whether DDI placebo was inert or not. But if we told people, if we acted on these results in our CPCR-07 trial and told people, well, this difference, lovely difference of seeing almost a halving in the progression to asymptomatic AIDS death and a halving in death rate, would be not due to active DDI, it would be due to the harm in DDI placebo. So in essence, as we've often thought, this is a great example of the tensions that monitoring committees can face. On the one hand, when I often call this individual ethics, where we're safeguarding the interests of study participants, where you might say, well, then let out the word that DDI placebo could be harmful, versus I might call this collective ethics, preserving trial integrity and credibility by ensuring that we're not prejudging results and that we're releasing results only when we have reliably answered questions the study was designed to address. So these are the issues that we'll continue to focus on, that we've been talking about to date and will continue to focus on today. These are, in essence, in my view, the main challenges that define or characterize what is the mission of the Data Monitoring Committee. So there are some fundamental principles, and Susan's nicely laid these out already in her presentation. We'll talk later on today about the importance of reducing prejudgment of interim data and why maintaining confidentiality is critical to that. And we'll also talk about why unbiased judgment is really critical, as Susan was alluding to. And we say, wait a minute, we have group sequential monitoring boundaries. They can enlighten us about when efficacy data are reliably answering questions the study was designed to address. And while they are extremely helpful and do provide considerable guidance, I bristle when anybody calls those stopping rules. They're not rules, they're guidelines. And even if the endpoint is highly representative of what patients care about, there are different analyses for the primary endpoint. There are secondary supportive endpoints that we could be looking at. There are safety issues. There are quality of trial conduct issues. There are issues regarding what we know from external data. And at the end of the day, while group sequential monitoring boundaries are very, very important guides, we need more than a statistician on the DMC to tell us whether the boundaries crossed. We need judgment. And when you think of the characteristics of a judge, 2 characteristics that I think about are that we want a judge to be well-informed, and we want the judge to be independent. And I always use it as an example. If Susan and Janet had a property dispute with me, and it went to court. And the judge was a sibling of Susan or Janet, I'm sure that person would be high integrity. And yet I would have a concern. That would not be independent. And so conflicts of interest doesn't mean a person has in any way a compromised character. It just means that their ability to be independent and objective is compromised. And so it means that, as Susan indicated, some of the really key principles that we have are that the data monitoring committee should have sole access. And as I mentioned, we'll come back and talk a lot about that. They should be multidisciplinary. And as Susan indicated, that's biostatisticians, clinicians, and potentially ethicists, epidemiologists, basic scientists, patient advocates. I've been alarmed to see an instance a couple years ago, first time I'd seen it, where someone proposed, and it was in a setting where the trial had an adaptive design built in, that will have only clinicians on the data monitoring committee. And I think it was from that same entity that the same issue surfaced again recently, where I think that same entity was proposing that there'd be only clinicians. I would have a serious concern that that would be multidisciplinary. We need surely the clinical insights that are critical to the specific disease setting, but we also need the quantitative insights that biostatisticians would provide in that multidisciplinary process. And then independence. And as she pointed out, we have a lot of people who are really integral to the clinical trials process, but they're not independent. So the sponsors, the investigators, those that develop the intervention, caregivers, regulatory authorities, would have conflicts of interest. And so the monitoring committee would be chosen in a way to address those potential conflicts. So Susan mentioned in a brief history, Susan mentioned that the concept of data monitoring committees first surfaced or was first put forward by the Greenberg report at NIH in 1967, where in essence, their ideas were to terminate early if the questions the study had been designed to address had been answered or couldn't be answered. And they noted this should be done by outside consultants. And as Susan and Janet mentioned, the concept of data monitoring committees then started to emerge more broadly in the late 1970s and early 1980s. We brought them into some of the NIH sponsored cancer trials. We noted, I remember having a discussion with Dave Demetz in the late 1980s saying, but you know, industry is increasingly carrying such a significant part of our research leadership and effort in clinical trials. Shouldn't we have data monitoring committees in industry-sponsored trials? And we knew that to have the influence and the guidance to do so, it was critical to have regulatory authorities on board with that concept. I remember Dave and I visited FDA, and there were very enlightened leaders there like Bob Temple, Susan Ellenberg, Jay Siegel, and others who were very influential in that process of developing guidances for data monitoring committees in industry-sponsored trials as well as in government-sponsored trials. And regulatory guidances then were developed not just by regulatory authorities, but by NIH and WHO. And it's led to what has now been a 30-year history, in my view, of remarkable implementation of monitoring committees where industry has done a remarkable job in incorporating those committees into their clinical trials. Okay, so let's press ahead. I'm going to start a number of examples that I want to give in addition to the one that I've talked about in 007, CPSR 007. So my experience with monitoring committees, one of those first experiences was in cancer, in the oncology setting, in the colon adjuvant setting. And I'll be talking about that a couple of times today. The colon adjuvant setting is a setting where we can actually achieve cures. If the surgeon makes a complete en bloc resection of colon cancer and has clean margins and there's no known metastatic disease, we're hoping we can call that a cure. And yet if that excised tissue has positive nodes, we know there's a 50% chance that there's undetected microscopic residual disease that will lead to recurrence and death within 5 years. And so the idea then was to use adjuvant chemotherapy levamisole in this case, or 5-ethylevamisole given in the first 5 weeks after surgery to try to eradicate that microscopic residual disease and, in essence, improve survival, or the secondary supportive measure, which would be recurrence-free survival. And so this was a landmark trial. It was one of the ones that was a first-time aggregation of NIH-sponsored separate cooperative groups that was merging the efforts of the Southwest Oncology Group, the North Central Cancer Treatment Group, the Eastern Cooperative Oncology Group, to do this, to enroll 1,000 people and follow them until we had about 500 deaths, where we had an O'Brien Fleming design analyzing the data about after every 125 events. So as was pointed out in Susan's presentation, the O'Brien Fleming monitoring boundary, if you're looking at it after every quarter of the information, would be basically using 4.2 standard errors, 2.9, 2.3, and almost unadjusted 2.0 standard errors. And so that monitoring boundary can be used for stopping early if we have established benefit, for ruling out benefit or establishing harm, much as Susan indicated. So in the case of the colon adjuvant study, where along the y-axis we're looking at where levamisole or 5-levamisole has no effect on the log hazard ratio, or if the hazard ratio is a 20% reduction or a 35% relative reduction in death rate. And the monitoring boundary, the upper boundary for stopping for benefit occurs if you cross any of these white arrows, ruling out equality, or stopping early for lack of benefit in the yellow arrows, ruling out a 35% reduction in mortality, or stopping early for harm, ruling out equality. And just really quickly to point out these characteristics, any of these boundaries, you could stop early for ruling out a 35% reduction. You could have a green boundary in between, stopping early, ruling out a 20% reduction, which is the threshold for statistical significance. Or you could wait and stop early only when you prove harm, when you rule out equality. And this is at the discretion of the sponsor. If I'm on the monitoring committee, I'm stopping the trial here for sure if we are establishing harm, but it's not an ethical imperative here to stop early simply because at this point we've ruled out major benefit. And so this is a decision that has to be made by the sponsor. This yellow boundary of basically stopping early when you're ruling out the alternative against which you have 97.5% power would be similar to a boundary if you were using a conditional power type approach, or using the idea that when in the trial do you have results that are sufficiently unfavorable that you're highly likely, if you continued, that you would hit a boundary for benefit. And this aggressive lower boundary has that property. And yet many sponsors might rightly say, okay, even if I am at this point halfway through and my results are fairly neutral and I'm no longer likely to hit a boundary for benefit, I still would like to continue. I've invested 90% of the resources in the trial. I would like to find out more specifically where I am at the end and only stop if I have really unfavorable results. And so you might use a green boundary here, stopping when you rule out a 20% benefit, or in fact, in some cases, wait until you've proven harm. So what happened in the colon adjuvant study, where we were looking at 500 deaths here along the axis, and here are the dates when the study started. It started in fall of 1984. It actually was fully enrolled after 3 years in fall of 1987. But the first quarter of the events didn't occur until spring of 1988. What you see right here is the O'Brien Fleming monitoring boundary in yellow at the 125 events, at 250, at 375, and at 500 events. So what actually happened, and this was, I think this was my first experience on a data monitoring committee. And in spring of 1988, we had our first formal interim analysis meeting with 25% of the events in hand. And at that point, because enrollment was only finished in the fall of '87, the data were, from survival perspective, still pretty immature. 5-year survival is kind of the benchmark for survival in oncology colon adjuvant settings. But we did have some really strong trends on recurrence. So we said, go on, but we were certainly anticipating that at the time of the next interim analysis, we may readily have definitive evidence. This time period as well in spring of 1988 was a very interesting time because that was at the point when we had a significant sense of concern with the epidemic of HIV. And there was a lot of effort at that point emerging around HIV to say, as some of our HIV advocates were saying, do we do body bag trials? Do we only do studies that actually look at survival? Should we be relying more on biomarkers? And in our setting here in oncology, we said, well, if you can delay the detection of recurrent metastatic disease, is that not a pretty good indicator of potential benefit? So this was the tension that we were experiencing at that point. And so in our novel, relatively inexperienced position for data monitoring committees, we decided shortly after this spring 1988 meeting, in the summer of that year, to have a very controlled, confidential meeting with 2 members of FDA who would have regulatory oversight if the next meeting led to a positive result, and 2 members of the sponsor for the trial. And they all pledged to maintain confidentiality if we shared data with them. So we did. We shared the data with them to let them know what we were seeing and to indicate that it was our intention, as we recommended, to continue the trial, but that we wanted to alert them that it was really potentially the case that we could be stopping the trial of the next analysis, so they would be in a position and be prepared for that outcome. Well, that pledge of confidentiality lasted 1 day. The next day, one of these 4 people, and to give you a clue, it wasn't someone from FDA, breached confidentiality, and that information was soon thereafter shared with former NCI Director Vince DeVita, who proceeded in the summer of 1989 to write a scathing editorial on how could a monitoring committee be continuing a trial when you have positive effects on recurrence. By the way, that's the last time. It was my first experience. I've been on 200 data monitoring committees. I don't think we've ever repeated this. We've learned from our experience that we don't share interim data with leadership and of trials and even regulatory leadership in nearly any setting because of the risks to trial integrity. So we moved on in the trial. The next analysis was to occur when there were 50% of the events. But at that point in time, there was quite a flood of events that came in, which happens to be the case in the colon adjuvant setting. People were now 3 to 4 to 6 years into the randomization. And so when we actually had our meeting, we had 300 events. We were at the 60% information point, not the 50% information point. Well, that's the beauty of the Land-Metz implementation of the O'Brien-Fleming, realizing you're really putting in place a continuous curve. And so, yes, at the 50% information point, it should have been 2.9 standard errors, and here 2.3. Land-Metz realized that at the 60% information point, you're using 2.6 standard errors, a significance level of 0.05. That was met as a one-sided p-value. That 0.05 was met by survival at that point, reinforced by now compelling evidence that, yes, this survival difference was being achieved by all indications by actually reducing, in a compellingly obvious way, the occurrence of recurrent metastatic disease. And so we decided at this point to release the data in the fall of 1989. And remembering that enrollment had ended in the fall of 87, NCI didn't sit on their hands for 2 years. After they enrolled our trial, they then spent the next 2 years up to the fall of 89 looking at 5-FU leucovorin against no treatment. And so one of the benefits of our releasing the data in the fall of 89 was that they now changed from a superiority trial of 5-FU leucovorin to a non-inferiority trial of 5-FU leucovorin against 5-FU levamisole. Another benefit of our release of these data in fall of 1989 is that at that point in time, FDA had rarely, in fact, achieved or granted regulatory approval for studies that hadn't gone to their completion point because they were appropriately very concerned about multiplicity issues. But given the careful structuring of this trial by a group sequential guideline with independent DMC monitoring, after this data was released, soon thereafter, FDA approval was granted of levamisole. So a classic case here of where the use of pre-specified monitoring boundaries with clarity of how multiplicity was addressed with independent monitoring by a committee was important to regulatory authorities. Now, this was a cake and eat it too setting. So while we had our cake of being able to improve or refine the design of new ongoing studies and achieve earlier approval of levamisole, the participants in this trial weren't crossed in. And it's unusual. Usually when you reach a conclusion for benefit, the control patients get the active. But in this setting, the active was adjuvant chemotherapy, which was strongly thought to only be beneficial if you're administering it fairly soon after surgery. And when we stopped this trial, all patients were already 2 years, and in many cases, 4 to 5 years post their surgery. So it did allow us to actually have our cake and eat it too, which was to see what long-term results were. When we stopped the trial, when we declared that the results should be released in late 1989, these were the results where we had marginal evidence out to 3 years, p-value one-sided 0.003 on survival. When we looked at it then 3 years later, that data out to 3 years was now out to 7 years, and we had a strong reinforcement that the monitoring boundary had led to a proper release of information to allow earlier access, several years earlier access, where those results were sufficiently reliable for the long-term benefit. Usually though, and it's a great lead into this slide on organizational meetings, a lot of these issues we could anticipate and should be discussed with the sponsor. I always say to the sponsor, I'd love to have you at the table with us when we are doing our interim analyses, because you folks bring such insight, as do the investigators and regulatory authorities, and because of independence issues, they can't be with us. So the organizational meeting is such a critical step that occurs ideally when you have a penultimate version of the protocol where the DMC can sit down with these other authorities and talk through these issues that influence in part what the proper monitoring boundary should be, but also other aspects of the trial design. So an example that I use, there's so many examples, but I was on a protocol or data monitoring committee in meningococcemia, which is it's a terrible infection in young kids that can be fatal or else lead to amputations in 10+% of these children. And there was an intervention called bactericidal permeability increasing protein, BPI, that was being studied against placebo. And in that setting, fortunately, they did recognize we needed to have interim analyses to protect these kids if early results were unfavorable or favorable. But if you were on that monitoring committee and you were not comfortable with that monitoring boundary, now is the time to have that discussion with the sponsor at the organizational meeting. So one example, I was asked by National Institute of Mental Health in 1991 to chair a monitoring committee for a trial of peptide T that was being given in HIV-infected patients to see whether peptide T would improve their cognitive impairment. And it was a traditional randomized control trial of peptide T against control. The problem that we had during the organizational meeting was at 6 months, these control patients were crossed into peptide T. There's always that sense investigators would like to recruit patients in and say, we're going to give you peptide T either now or later. An argument was, well, peptide T has not been proven to be effective. A cross-in design presumes it's effective and just is randomizing to you when you get it. And we're saying the compromise is you're losing the ability to understand longer-term effects. So we argued in the organizational meeting that the control should not be crossed in at month 6. Furthermore, the study was designed so that those people that stopped randomized therapies, they called them dropouts, would be left out of the analysis. We said, wait a minute. That's informative missingness. Everybody should be followed. It should be an intent to treat analysis. And thirdly, related to what Janet and Susan have talked about, the sponsor, NIMH, said, we're going to give you safety data alone because we're not going to expect you're going to stop the trial early for efficacy. We said, fine, we get it, that we won't stop early for efficacy. It's not irreversibility mortality. Yet to understand safety, we have to understand it in the context of benefit to risk. We have to have efficacy as well. So what happened after we made those requests? The sponsor agreed and said, we're going to give you efficacy as well as safety. The sponsor agreed that we should do intention to treat. But the sponsor said, well, wait a minute. We have an agreement with the investigators that all enrolled people will get peptide T. So in spite of what you're saying, Data Monitoring Committee, we're crossing people in. Well, we're advisory, they're decision makers. If this were an ethical imperative, we might resign. But that was their decision. It wasn't an ethical imperative, so we proceeded. And so it gives you a sense of the nature of interactions that should occur at the organizational meeting. We provide advice, the sponsor makes decisions. At that organizational meeting, we've talked about the charter, we'll talk more about it. That should be refined and finalized. At this organizational meeting, as Janet so enlightened us yesterday, it's critical for the monitoring committee to be confident that the independent statistical reporting group is going to give them information they need. We have to be agreeing on the content and format for that information we're going to get, and we have to be confident that the entities here are going to be giving us information of high quality. So these are all things that happen at the organizational meeting. Okay, does that mean then that once the organizational meeting is done, we wait for interim analyses? No, not at all. We meet on a regular basis, even before formal interim analyses, having what we call early safety trial integrity reviews. And this colon adjuvant 0035 trial is a classic example. As we've already shown you, the first interim analysis didn't occur until almost 4 years into the trial. And by that time, the study enrollment shown in orange was already complete. If we'd waited for the first interim analysis for our meeting, we would have missed a very important oversight that monitoring committees should have early in the trial. Safety results can occur early, and we have to be monitoring. We should be monitoring the overall ability of the trial to achieve its intended objectives. So quality of trial conduct. Is it enrolling in a timely way? Critical issues, 3 critical issues. Are you getting proper adherence to the intervention? Are you getting proper retention? And are you getting the proper event rates? Something we've talked a lot about in our ongoing HPTN 083 and 084 trials in pre-exposure prophylaxis in HIV. Are we getting populations that will be sufficient to give us our targeted numbers of events? So these are issues that need to be monitored on an ongoing basis by the monitoring committee, and readily, as shown in this previous example, these are issues that can readily be of critical importance even before you have the time of the first interim analysis. Okay, so then we have the formal interim analyses, and we think very often at interim analyses about the issues of stopping early for benefit, or stopping early for lack of benefit, sometimes called futility, or stopping early for established harm. But at interim analyses, to be based on experience, this isn't what typically happens. Termination of a trial isn't what typically happens at an interim analysis. It's a possibility, but in my experience, in a couple hundred trials that I've monitored on DMCs, 10%, 15% maybe have stopped early, and typically they will have 3 DMC meetings. The Precision trial, that was an 8-year trial, we had 20-some DMC meetings, but typically you have about 3. And so that means that in 95% plus of all DMC meetings, in my experience, the recommendation is to continue, but with important aspects of how to conduct the trial in manners to maximize addressing ethical, safety, and trial integrity issues. So one more example of these issues around early termination. I was on the data monitoring committee in the normal hematocrit trial that was done in end-stage renal disease. And in end-stage renal disease, patients are at high risk for death and MI. And there's a strong sense that that risk for death and MI is influenced by their hematocrit level. And so we give erythropoietin-stimulating agents. And standard dose ESAs are partially effective in normalizing hematocrit. And yet when you look at people getting standard dose ESAs, what you see is that those that have, in spite of standard dose ESAs, inadequate levels of hematocrit tend to still have high levels of death MI. And so the idea is if standard dose ESAs are good, maybe high dose ESAs are even better. So a randomized trial was done, the normal hematocrit trial in randomizing in this way. And we randomized 1,200 patients. The study was designed to be followed until we had about 720 death MIs. And in this case, death MIs are almost always deaths. So about 720 deaths. So we were halfway. We were exactly halfway. We had 360 of those 720 events. And at that halfway point, what we saw was it worked. High dose ESAs gave much better control normalization of hematocrit than standard dose ESAs. And by modeling, one would have expected that that would have led to about a 25% reduction in death rate. Yet, the data said, no, it was actually a 30% increase in death MI or death rate. There were about 40 excess events, not 40 prevented events, because biomarkers are often unreliable surrogates. Correlate is not a surrogate make. When you normalize hematocrit, you don't just achieve the benefits of normalized hematocrit, you have off-target effects. In this case, we think it was an increased incidence of thrombosis of vascular access sites. So we were right here on the monitoring committee at the halfway point, where if we had been a 25% reduction in death MI or death, we would have crossed the boundary for benefit. If it was anything less than neutral, we would have ruled out benefit. We would have known we weren't going to cross this boundary. At least we would have ruled out a 25% reduction. The actual estimate that we had wasn't up here as the surrogate would have told us with a 25% reduction. We had 40 excess events, and we had blown past the boundary for the stochastic curtailment boundary, or the boundary that would basically say we've ruled out major benefits or ever get positive results. In fact, we had not only ruled out a 25% benefit, we'd ruled out even a 13% benefit, which is what it would have taken at the end to be statistically significant. We'd even ruled out a 6% benefit. We'd almost ruled out a quality to prove harm. And so our data monitoring committee recommended termination. Our judgment was to terminate. By the way, if you go back to the New England Journal and review the abstract written by the sponsor, who still believed that normalization of hematocrit should have been a good thing, they said the monitoring committee stopped even though a boundary wasn't crossed. I beg to differ here. We ruled out any level of benefit, which if real, could be clinically relevant, even a 6% benefit. And I've often wondered if the authors of that would have liked to have joined the trial at this point, where we had a suggested 30% increase in mortality, ruling out any meaningful level of benefit simply to prove whether or not we actually were killing people. So this was the judgment by the monitoring committee that this was, in fact, definitive evidence to rule out benefit and nearly prove harm. So we talked in the last couple of days about the important issue of where monitoring committees fit into what I would call a partnership of responsibilities. And this is really important. The monitoring committee is key, but many other entities are key. Institutional review boards, IRBs, are really central, especially at the point of approving the ethics and science of trials, as are regulatory authorities. And regulatory authorities are also key during the course of studies, particularly for looking not just at SAEs, but suspected adverse reactions that are serious and unexpected. By the way, an example of this, lamericoxib, a COX-2 inhibitor, was being studied. And in that study, if the monitoring committee was seeing MACE events, macrovascular complications, cardiovascular death, strokes, MIs, those aren't SUSARs. We know that COX-2 inhibitors have already been reported to have some risk of effects on macrovascular complications. But what was a SUSAR in that case was there were cases unexpectedly of fulminant hepatic failure, which hadn't been expected, and that would be characterized as a SUSAR. By the way, an excellent paper on this was written by Janet and colleagues in 2015, talking about safety assessment committees, and there have been subsequent discussions about how data monitoring committees can play a role in that as well. So in addition to these entities, sponsors and investigators are critical. They maintain the decision-making responsibilities about the trials. We on a DMC are advisory. They make decisions, and caregivers make decisions about primary care responsibilities for participants in trials. Yet all of these entities rely on us in a significant way on a DMC because we have sole access to emerging data on efficacy and safety. And as we've talked about and will continue to talk about, we need to be trustworthy. We need to be entities that can be trusted by all of these other entities who normally would have access to these data, but are not having it in these cases to protect trial integrity, and they're relying upon us and our judgments. So it's very much a partnership here between the DMCs and all of these other entities. So what I'd like to do in spending the second half of this first segment and all of the second segment, so 3/4 of my time today, I would really like to spend talking about something that Dave DeMetz kind of inspired me, or certainly confirmed my own sense about. And that was about 6 or 7 years ago. Dave sent me a slide that basically said this exact sentence up here. In opinion, he said, the DMC process for monitoring randomized trials is not better than it was 10 years ago. And while Susan and Janet and Dave and I and so many others that have been involved in this process are incredibly appreciative of the positive consequences from the implementation of data monitoring committees, all of us are aware that we face a challenge that, particularly as this concept has grown and has been much more widely implemented, to achieve its benefits, it needs to be done in a proper manner. And so when I saw this quote from Dave about 7 years ago, I called him and I said, Dave, this is exactly matching my own experience. And where my concerns were and Dave's were that what we were worried about is whether there were emerging practices that were compromising the independence of monitoring committees. And we believe that it would be very important to develop best practices and operating principles, or to reinforce those best practices and operating principles for effective functioning of data monitoring committees. And so what we did is we proceeded in June of 2015 with an expert panel, and a number of the members of that panel are here. We had remarkable contributions from so many, like Janet and Susan, on that panel. And it included stakeholders from academia, from industry and government sponsors and regulatory authorities and legal. So it had the DMC stakeholders from a wide array of different disciplines and expertise. And our goal in that process was to basically arrive at a consensus and to publish an article on best practices. And we, in fact, did eventually publish that article almost 2 years later in 2017. Why did we wait 2 years? Well, that article, in my own experience, I don't know about others, but I spent more time on that article than any article I've ever published. I think I logged over 250 hours time, and that wasn't counting the enormous amount of time that others put into it. But our goal wasn't just to write another article. Our goal was to identify the critical issues that were influential to best practices, arrive at a consensus where we truly had consensus amongst ourselves about what it was that we wanted to advocate, and then to state it with clarity. Every sentence in that article was debated at great length, but we felt it was critical if we were going to come forward in a way that would be positively influential in promoting best practices. And so we talked about probably 10 to 12 critical issues. A number of them are here. The importance of experience and training, indemnification, why data need to be current, confidentiality issues, ways to enhance DMC independence through proper meeting formats, through a charter, the consensus process, the contracting process, and very importantly, as Janet was talking about yesterday, the independent statistical reporting group. So all of these were the topics that we had discussed at length. And I want to go through these issues as we had discussed them one by one. And so I'm going to start with the issue about training and experience. So a survey had been done that was presented to us in this working group, and it was really informative. And I think it matched a lot of our sense from our experience. And that is, there's not a lot of formal training opportunities for people when they are invited to serve on a DMC. That certainly was true for myself. I'd never had any training when I first served on a data monitoring committee. And the survey here said only 8% of people said they had. Yet nearly all of them said they wished they'd had training before they were on monitoring committees. And training is really key. It's especially critical for, many of us would say, the DMC statisticians, and certainly the data monitoring committee chairs need to be well trained in the DMC process. And some of the critical aspects that they need to understand as a DMC chair is that, of course, in the closed session where it's just the chair and the committee members, they'll be in a lead role. Many of us believe they should be in the lead role, even in the DMC open sessions where the sponsor is there to be taking charge and leading us through that process in an independent way. And in the closed sessions, it shouldn't be a passive approach where the chair sits back and says, does anybody see any problems? If not, maybe we'll have a short meeting. This chair should lead or ensure that the committee is led through the key findings in the report. I loved it was Susan or Janet was talking about the concept the last couple of days about a chair who assigned certain sections to various members. That's a fantastic idea because it does engage the entire committee. You know when you show up at the meeting that everybody has cracked the book and has looked at it in some depth. And as we've seen in Janet's discussions, the role of the independent statistician is critical, and their experience and training is also really key. So, okay, I get it. We need training options. We need them for members of the DMC, particularly the chairs and the statisticians. We need them for the DMC meeting coordinators. We need them for the independent statistical reporting group. They all need it. And so an obvious way is to find a textbook. Shamelessly promoting one of these textbooks. We need textbooks, we need articles, and we need, like what we're doing here in the 3 days, web-based lectures, interactive courses. These are all very significant steps in the process of training colleagues in the DMC process, either as for members or as people that would be as independent statisticians, or for people that would be sponsors, engaging them, or people that are in fact just understanding what the data monitoring committee is doing and what it takes for them to be in a trustworthy position. So these are all really critical. But an example that I use is, if I was going to fly a small plane, would it be adequate for me to carefully read every manual that I could find and every textbook and go through all the lectures on what to see in flying that small plane? Or would it actually help for me to have some experience with an instructor flying the plane with me? I surely think so. And so what we've argued in this consensus best practices group is that we should be advocating apprenticeship models that enable data monitoring committee members to be involved. And so if we have, let's say we're on a committee right now in COVID, in hospitalized COVID patients, I could imagine that that committee should have a clinician who would be a pulmonologist, and one who's a cardiologist or hepatologist, and one in infectious disease. And it surely should have a statistician. Well, collectively, those 4 people, including the chair, needs to have experience. Is it necessary for all 4 of them to be experienced in the DMC process? It's necessary for all 4 of them to be knowledgeable in the science that they represent. And yet, we actually should have some heterogeneity where 1 or more of these people don't have experience in the monitoring committee process, because that is, in fact, expanding the number of people who do, while we're still sure that every monitoring committee has experience. And by the way, that's going to work for the clinicians, because you have a pulmonologist, a cardiologist, and an infectious disease doc. So 1 or 2 of them is experienced, 1 or 2 of them isn't. But if you have only a single statistician, and I've said 2 slides ago, the statistician should be among the people with experience, then we're perpetuating a situation where we have a fixed number with experience. And so we do need settings. I think I've heard that Seattle Genetics is among them. We need companies willing to actually put a second statistician on a data monitoring committee, specifically with the idea that they're both very knowledgeable about the scientific process, but one of them has DMC experience and one doesn't, and this will help us build on that experience too. If we had a DMC with 3 clinicians, a cardiologist, a infectious disease doc, and a pulmonologist, I don't reimburse only the ones that have been on DMCs before. I expect every one of them to be a major player in the functioning of that DMC. And if we had an experienced statistician and an excellent statistician who knows the statistical issues but doesn't have DMC experience, my view is they're full members of the group. They need to be fully reimbursed. And the apprentice concept is just that, yes, this is now engaging the inexperienced DMC person who's very experienced in the science in general to be now and experienced in the process. Okay, I'm going to jump ahead here because I know we're going to be running into some time constraints. Indemnification is another key issue. In this particular issue, the fact that there are liability issues for data monitoring committees is something that many of us don't think about. I will admit, I've been involved in DMCs as a member for 35 years. It was 15 to 20 years before it even dawned on me, if we're doing our job well, and I know we always do, not to worry about litigation. And by the way, in the early days, probably not many of us knew about data monitoring committees. Well, nowadays, data monitoring committees are a much more known entity. And so the issues about liability risks are real. There are multiple sources of these liability risks. Just to give an example, one that I like to use, if you have a platelet inhibitor and you're monitoring that trial, and into that trial, you would see 5 excess hemorrhagic strokes in the platelet inhibitor. But when you look at the data, you also see 5 fewer ischemic strokes, 10 fewer MIs, and 5 fewer cardiovascular deaths. And so the net effect is clearly positive. And I wish I knew who was going to have the hemorrhagic stroke, but I don't. And so you make its benefit to risk. Again, why knowing benefits are so critical in judging risks. So you recommend continuing. And at the end of the trial, the same pattern persists. Now with sufficient numbers that the study is clearly a positive result that you've reduced major morbidity and mortality. But the data were released, and the family member of someone who late in the trial experienced a hemorrhagic stroke sues the data monitoring committee because they knew that this risk was there. Well, it's clearly a case where the monitoring committee made the right recommendation, and yet they were at risk. And so there was a meeting that was held. Dave Demetz led it. Many others participated. The article was published in 2004 that presented the recommendations for that meeting, recommending that it's critical to have indemnification for data monitoring committee members. And the academic lawyers were very wise in pointing out that when you have indemnification, industry sponsors may well wish to have escape clauses like negligence, which is not a proper escape clause, as we were told at the time. Ask those lawyers if they have a negligence escape clause in their auto insurance. So the escape clauses should only be more significant willful misconduct or fraudulent acts. What's interesting is the night before the meeting that was held that led to this article, it was a consensus meeting and NIH lawyers had been invited. They pulled out the night before that meeting. And they pulled out in part, at least as reported to Dave, was that they were aware, they'd become aware, or they were aware of the Federal Anti-Deficiency Act in Ellenberg, Fleming, and Demetz. This is discussed on page 393. And this act prohibits the government from indemnifying non-government employees. So NIH lawyers said, we can't indemnify people who aren't, let's say, special government employees with FDA or otherwise government employees. And it was interesting. I was thinking for 20 years, I had served on mostly as a pro bono act, monitoring these 50 trials for NIAID, never dawning on me that if we were sued, that NIH wouldn't in fact be able to defend us or support us. So this is a very real issue. When in industry, when I get contracts sent to, in fact, engage as a data monitoring committee members, very frequently, particularly if these are developed by a CRO on behalf of the sponsor, it addresses indemnification by saying, the DMC members will indemnify the CRO, or we will indemnify the sponsor instead of the opposite. Patricia Trescas, who I've never met, a lawyer from Virginia, wrote a really fascinating article where she says that there ought to be congressional legislation requiring all sponsors to indemnify DMC members and to empower them to select and retain their own counsel. And by the way, this issue is being increasingly effectively addressed where DMC members are indemnified. It may take, I've had 4 to 8 hour negotiations in some settings with sponsors to achieve this, but having proper references and all certainly help. Although I've never succeeded in being able to do what Patricia said, which is to achieve the ability to select and retain my own independent counsel. We have made the argument though, and I think it's a true one, that indemnification is not just important for the DMC member, it's important for the sponsor. There have been in recent years where indemnification risks are more understood. There have been a couple settings where I've been on monitoring committees where 1 or 2 of my colleagues have said, you know, we're making the right recommendation here to go on, and yet we could be at legal risk. I mean, there are ways that it could be misinterpreted and we could be at legal risk. And I would greatly regret if we were ever in a position where we terminated a trial before obtaining reliable evidence because the monitoring committee was fearful that they could be inappropriately, but nevertheless sued, and that the sponsor would in fact not be protecting them. So it's really for the sponsor's protection as well. So one other quick thought before we break, and that is something to build on what Janet was saying yesterday, and that's the importance of currentness of data. So I'll give an example that highlights why this is really important, that a monitoring committee, when it meets, should have information as current as possible. This is one of those many 50 trials that I was mentioning that I had a chance to monitor over the 20 years from 86 to 2006 by ACTG or CPCRA by the NIAID cooperative group. It was the AIDS Clinical Trials Group 019 trial. This was really early on in HIV/AIDS research. It was a study started in 1987. It was in asymptomatic patients, although their CD4 counts had dropped below 500. Placebo was current standard of care, but this was like walk before you run. This was the first step. Would AZT or zidovudine in those asymptomatic patients started when their CD4 count dropped below 500 be to their benefit, where benefit was either symptomatic AIDS events, capsular sarcoma or opportunistic infection, or death. And we had an interim analysis 2 years into the trial. This is what the data showed. We had over 1,000 patients enrolled, and we had 51 patients that had clinical endpoints, that had symptomatic AIDS events or deaths that had occurred. And the data were really encouraging. We kind of think, it kind of feels like COVID at the beginning of COVID. We have nothing and we're trying to find something. This is where we were in HIV in 1987. We had nothing. We were trying to find something. And this was something. This was an indication of a 2 to 3-fold reduction in these major symptomatic death events. And yet we looked at the data that we had on August 2nd, and the data lock was on May 10th. And we said, the world is waiting to hear from us. We're going to make a recommendation based on 51 events, pretty small number of events, when there could be a lot of other events that emerged between these dates. So we said to the independent statistical reporting group, please don't update the whole report. Update the primary endpoint, make it current, and get back to us as quickly as you can. And they did a great job. They got back to us in 2 weeks. In 2 weeks, they had current data. Did it matter? Yeah. The 51 events, there were 23 others lurking out there that that hadn't been reported to us. And by the way, it was a different pattern in those 23 other data events. Yet, nevertheless, with these updated data that now gave us basically a halving in the event rate, we did cross the boundary, the O'Brien-Fleming monitoring boundary, and we did recommend that the data be released to the sponsor. But that update mattered. because this is what the data looked like prior to the update. This is what it looked like. It looked like, wow, this could be a durable long-term reduction in the risk of progression to symptomatic disease or death. The updated data told us something that more experience would absolutely confirm. No, single agent AZT wasn't curative and certainly wasn't long-term benefit. It was about a 6-month lag. So that data, that update mattered. And I remember flying home, 2 days later, flying home, this result was on the front page of the Washington Post. This was like COVID is today, trying to find something that matters. This really mattered. And it was really critical for us to have current data. And so principles that we've had is we need clinical cut dates, we need data lock dates, we need not a passive approach, we need a proactive approach. So if the data monitoring committee is going to meet on April 1st, then I want to know that anything that's happened to somebody 6 to 9 weeks before that, anything that's happened to somebody by February 10th, I'll call that the clinical cut date, needs to be captured and in the report. The report is based on the data that are locked. That data lock date should be March 10th. So clinical cut date February 10th, data lock date March 10th for an April 1st meeting. That gives the independent statistical reporting group 3 weeks, basically 2 weeks to generate analyses and reports. By the way, if I'm on the DMC, do not send me the report 3 weeks in advance. I'll be angry with you because I'll know you could have given me 2 weeks of more current data if you sent it to me a week in advance. So it's my job on the DMC to know that in that 5 days before a meeting, I'm going to have time carved out to look at this report. And so effectively, what we want to be doing here is proactive, certainly leading up to February 10th and between February 10th and March 10th, get everything by the time you lock the data current through that date of February 10th. And by the way, the SAE data and the non-validated endpoint data are also presented to us at that data lock date. And this is key in so many ways, as we've just seen in the ACTG example, for having a more reliable insight. It's also key in distinguishing completeness and currentness. If I don't have data, is it because it's just not current, but it's being collected, or it's because it's actually never going to be collected? And I can't distinguish permanent loss of follow-up or completeness issues if my data aren't current. So this is a key issue that Janet talked about yesterday that we reinforced as well in this best practices group. Okay, so I wanted to spend, since we have 5 minutes, it's not going to give you a lot of time here for deliberation, but I do want to go back to the example because we need your help. Everybody on this call, you're on the monitoring committee for CPCR 07, and this is the data that you see. And how can you, folks, how can you allow continued randomization when you have 17 deaths on a placebo, DDI placebo, compared to 2 on DDC? But conversely, are you going to be guilty of over-interpreting these early data, making a change that could, in fact, severely compromise the integrity of this trial? So you're with us on the monitoring committee. What should we do? Or what else would you want to know to help you decide what to do? Here, Susan Ellenberg was at Division of AIDS. And so she was part of the team that worked together with CPCRA and ACTG. And as I understood, Susan had raised a very good question when the study was being designed. And that was this DDI placebo wasn't just a sugar pill. It was the actual DDI pill, but taking out the DDI active ingredient, but leaving everything else in, including a buffering component, et cetera. And that she had said appropriately, gee, is that truly going to be inert? So is this truly inert? That's another thing that we could think about. But there's another thing, and if you had more time and you haven't, but you had more time to look at this, not only when you look at the DDI placebo against the DDC placebo, is this difference of 17 to 2 attributable to a high number of deaths on DDI placebo? Look, it's really very much attributable to a low number of deaths on DDC placebo. if this is real, 17-2 driven by DDI placebo, then you're trusting that DDC placebo has a 1/4 the death rate of the DDC active. That seems highly implausible. So bottom line is there was no intention ever of doing this analysis. This was intended to be looking at 337 against 340 against 344. This was an artifact. I mean, it was a bonus that we actually got this because of the clever design to be able to reduce the number of placebo pills. And so one has to worry hugely about random high bias, about data-driven hypotheses. So in order to let you go to break, I'll explain what's at the top. We actually had an easier way out. Our monitoring committee for CPCRA 07 realized that there was a companion monitoring committee for an identically designed concurrent trial going on in Europe called the Delta trial. So we, on our monitoring committee, contacted our sponsor, NIAID, and said, NIAID, I won't tell you why, but will you contact the sponsor for the Delta trial and see if the two sponsors will allow our monitoring committees to share data? Fortunately, enlightened, they did. And when we saw the same exact designed ongoing trial data shared to us from the data monitoring committee for the Delta trial, this difference didn't exist. And so we had an increased sense of confidence that this could readily be random high bias. We hence recommended continuation after November of '93. When we finished the trial, these were the final results where we had fivefold the information from our trial. These differences in AIDS events disappeared completely, meaning that after this interim analysis, there were 40 more of these events on the DDC placebo than the DDI placebo. The difference in symptomatic AIDS death events disappeared. And even for death, there were 6 excess deaths on DDC placebo thereafter, so the final difference was 9. And so it brings up again the challenges that a monitoring committee faces as it is balancing individual ethics and collective ethics, realizing that we'll look at totality of the data, but when you have a data-driven hypothesis, this was entirely a data-driven hypothesis, we have to worry hugely about random high bias. And in this case, fortunately, we had the ability to see the results from the Delta trial as well to recognize that in fact, this difference was almost assuredly at random. So I want to pick up where we left off. We had talked in the earlier part of the session today about the mission of the DMC, saying it's individual ethics and collective ethics. It's balancing the first and foremost safeguarding patient interests, participant interests, but also protecting trial integrity and credibility. And so as Susan had pointed out, critical features and principles are maintaining confidentiality, having unbiased judgment, and that includes principles of multidisciplinary representation and 2 issues now that I'm going to talk a lot about in the rest of today's presentation, the importance of sole access and the importance of ensuring independence and protecting independence. So we've talked about training and we've talked about indemnification and currentness of data. I'm going to spend a fair amount of time here now in this last session today addressing issues of confidentiality. And so to give you some context for this, in 1995, I had given a presentation to colleagues in New Zealand and to the Health Research Council, which is their version of, in the US, the NIH. And shortly thereafter, the Health Research Council of the New Zealand government decided to put in place an independent data monitoring committee to basically serve as the monitor for the Health Research Council sponsored trials. And I had the privilege of being invited to chair that committee for 10 years. So I would fly down to Auckland for our meetings from 1995 to 2005. And they did really impressive things through the HRC. One of the trials that we monitored was a study in colorectal cancer, but it was in the adjuvant setting, But unlike before, where the other studies I mentioned were the 5-of-levamisole, that we're looking at using adjuvant chemotherapy as a way of improving survival in those patients. The purpose of this trial was to look at how can we optimally do the surgical procedure. So the standard approach was an open colectomy, and the study was looking at a randomization of these patients who were going to get surgery, hopefully for curative intent, for their colon cancer, to be randomized to an open colectomy versus a laparoscopic assisted colectomy. And the intention of the laparoscopic assisted approach would be it's less invasive, and that hopefully will lead to less hospitalization days, less cost, less pain for the patient. And so the study was well conducted, was underway. We were monitoring it regularly. It was a long-term study. And we got several years into the trial. And because of the size of the trial, the New Zealand government had engaged not just New Zealand surgeons, but Australian surgeons to be able to get a broader and more timely result. And as we were then many years into the study, some of the Australian surgeons approached the Data Monitoring Committee indicating that we, by that time, had fully enrolled the trial, so we really had complete data on the short-term endpoints of did the laparoscopic procedure give us less hospitalization days, less cost, less pain, and they wanted us to release those data, even though the critical feature of the trial was also to do non-inferiority of the laparoscopic procedure to open colectomy regarding survival, to make sure that this laparoscopic procedure, while being less invasive, wasn't leading to the critical feature of being less effective in getting complete en-bloc resections and prolonging survival of patients. And it was still ongoing as to whether or not that longer-term result of non-inferiority would be achieved. So the monitoring committee that I was chairing argued that to release information on parts of the potential benefit without understanding totality of benefits and risks could be biased, and we believed that such a breach in confidentiality could compromise the integrity of the trial, and argued that the data should be kept confidential. We were supported by actually the New Zealand surgeons, and we were supported by our sponsor, which was the Health Research Council in New Zealand. And yet there was quite an ongoing discussion about this. And the Australian surgeons persisted in really wanting to get access to these data. And we said, well, write down your arguments. What are your specific arguments? And here were their three particular arguments. The first, and I'll cover these individually. The first was the Australian surgeon said, early release of interim data could help increase the enthusiasm of participating investigators, particularly in studies that are somewhat prolonged. Well, to address this, interestingly, the United Kingdom Health Technology Assessment Program had commissioned a group called Data Monitoring Committee's Lessons, Ethics, and Statistics Study Group, or Damocles. And their intention was to investigate existing processes and identify ways of improving DMC processes. So I might say establishing best practices. And one of their conclusions when they reported out their results in 2005 was that the current prevailing view is that the trial investigators should not see unblinded interim results. And the argument that releasing interim data would aid enthusiasm and accrual, which is the argument of the Australian surgeons, they say is false. And one of the sources of their insights for this recommendation is a study that Susan had alluded to yesterday, that we had conducted back in the 1980s. And so for context for this, I was serving on NIH cancer cooperative study groups, and in particular the North Central Cancer Treatment Group when I was at Mayo Clinic. I was part of that group in 1977 when it was initiated, and I was there through the next 7 years, 77 to 84. And we brought in, early on in that process, our cooperative group brought data monitoring committees and it put them in place. In cancer studies up to that point, results were routinely reported at every meeting, at every steering committee meeting. So every 3 or 4 months, you would tell everybody everything you knew. And we broke that pattern by arguing that there should be monitoring committees in place to serve in the mission of the monitoring committee, including to maintain confidentiality. In 1984, after 7 years of this process, I had moved from Mayo Clinic to University of Washington and left the North Central Cancer Treatment Group and joined the Southwest Oncology Group. That had just been established, their stat centers had just been established at UW with John Crowley at the Hutch and at UW in charge of that stat center. So I joined them shortly thereafter in 1984. And I approached John Crowley and Chuck Holtman, who was the head of SWOG, and said, would you folks entertain the idea of implementing data monitoring committees in SWOG? And John and Chuck were both very supportive of the idea, but said, you got to convince our steering committee. So at the next steering committee meeting, I presented the concept for data monitoring committees. And I'll never forget, one of the senior influential clinicians said to me, what? We're going to have our regular meetings and you're not going to tell us the current data that are evolving in our trials? And I remember forgetting, he said, what are we going to do? Play tiddlywinks at these meetings? He said, at least you have to show us scientific evidence for why we're going to do this. And I thought that's a very valid request, very valid. So Stephanie Green had joined me, had also had transferred from North Central, from Mayo to University of Washington, Hutch to SWOG at the same time. So Stephanie and I worked together to basically do a comparison of the experience over the years of 1977 to 84, during which time North Central had limited data, interim data to DMCs, but where SWOG had reported all their data. And so we identified 10 major North Central trials and 10 major SWOG trials that were done during that 7-year interval that were colon adjuvant studies, breast adjuvant studies, lung adjuvant studies. And we compared what happened in the setting where data were kept confidential to a data monitoring committee versus where they were routinely reported at every meeting. And in these 10 trials, it's interesting how you forget how the world used to be 40-some years ago. But what happened, not too uncommonly back in that era, is that you would ramp up enrollment, which we do today. And nowadays, when you ramp up enrollment, it kind of stays until you're done. Back in those days, in 5 of the 10 trials, the enrollment ramped up and then had a precipitous drop over time. It didn't happen in any of the trials in the North Central Cancer Treatment Group where data were kept confidential. That prejudgment didn't happen. And in these 10 trials, 9 of them were completed at the time that Stephanie and I did this overview. And of these trials that had precipitous drops, 2 of them had been terminated early. Not actively terminated early because they'd answer the questions they were designed to address, but passively terminated early because this precipitous drop was so large, so strong, that we were never going to be able to finish them. Well, one great thing back in that era, when you reported everything all the time, is that we could publish our results every year. You could have a 4-year trial, you could publish the results on an ongoing basis. And yet 2 of those publications, when the studies were ultimately in 2 of the trials, those early publications were very inconsistent with what the final results indicated. And so this concept of maintaining confidentiality of emerging data was really important to avoiding the risk of prejudgment, in turn then increasing the ability to get timely enrollment, to complete studies in a reliable way, to get reliable results, and to reduce this risk of reporting out early results that are misleading. And this issue came up in the last couple of days with Susan and Janet. Another really important feature is if you're doing a trial over several years, there will be external evidence that emerges. Can you use that external evidence to refine the design of your ongoing trial? In many cases, you could, but only if the people making that decision to refine the ongoing trial are completely blinded to the results of that ongoing trial. And so another benefit of maintaining confidentiality is it's empowering study teams to have the ability to refine the designs of their confirmatory trials based on emerging external evidence. Well, the Australian surgeons also said that early release of data will provide more timely access to reliable results. Maybe, but maybe not. One example of maybe not was the CPCRA 002 trial. So this was even earlier than the 007 trial I showed you before. This was a study done very early in the HIV/AIDS arena, a time frame in 1990-91, where at that time we had AZT. Now we were trying and building toward other therapies. We had DDI as a proven antiviral that could be used if you became intolerant or you failed AZT. And our goal here was to see if we could broaden our arsenal of effective antivirals that we could use, antiretrovirals that we could use in settings where you no longer could tolerate or get benefit from AZT. Yes, you had DDI, but the question is, would DDC be an appropriate alternative? Would it in essence be similar to DDI? Or as we would say today, could you formally establish that DDC would be non-inferior to DDI relative to survival and relative to the primary endpoint of progression to symptomatic AIDS or death? And for the analysis here, to assess whether DDC was non-inferior to DDI on AIDS progression death, it would take 243 patients having events. And so the study was analyzed after equal increments of information, roughly after each 60 events, 3 interim analyses and a final analysis. So this slide basically shows you the essence of the primary efficacy data that we were monitoring over the couple years when we were monitoring the CPC Area 002 trial. And what you're seeing here along the x-axis here, what you're seeing along this left-hand side here are the dates of our meetings. We met on August of 91, in November of 91, in February of 92, and then although we were meeting quarterly, the next meeting was actually 6 months later in August of 1992. And what we were looking at here along this x-axis, what is this scale for? It's the scale for the primary endpoint, which is the rate of progression to AIDS and death. So it's symptomatic AIDS events and death events, and that relative rate on DDC relative to the standard of the active comparator DDI. And we hoped that the experimental DDC would be similar to DDI. So we hoped truth would be around 1 for the relative rates of progression to AIDS death. It'd be lovely, in fact, if the hazard ratio were in the positive direction, 0.8, where DDC would be even better. But a positive result would be if the estimate was sufficiently favorable that you could rule out the non-inferiority margin of a 25% relative increase in AIDS death on the experimental DDI against the standard of care, experimental DDC against the standard of care DDI. So what did we see? What information evolved? In August of 1991, we hit the point of having the first 25% of events, the first 40 events roughly, wasn't looking too good. There were 39 patients on DDC that had AIDS events or death events compared to half that, 19 on DDI. So our estimate was not unity as we hoped. It was a doubling in the rate on DDC versus DDI. And while we hoped at the end that we would be the same with a confidence interval ruling out that we were 25% worse, here our estimate we were double with a naive 95% confidence interval that ruled out that we could be as good as 25% worse. So, gee, that should be enough to stop this trial as a failed study. No, the monitoring committee recognized that there are multiplicity issues here, including the fact that we're testing at multiple points in time. So we were using, as Susan was pointing out, the O'Brien-Fleming monitoring boundary. That was 4 standard errors, 4.2, 2.9, 2.3, and 2.0 standard errors. So we were using 4 standard errors at this point. And when you use, this is 1.96 standard errors would tell you, okay, we're ruling out that you could be as favorable as 25% worse. No, the O'Brien-Fleming said, no, these data actually, when you adjust for multiplicity here, still haven't ruled out equality. Well, the O'Brien-Fleming monitoring boundary turned out to be right here, because as we then progressed in subsequent quadrants of the information, the estimate for the relative risk on AIDS death became eventually neutral, equal on DDC to DDI, ruling out 25% worse, being a positive final result. The final result was positive. It gave us another antiretroviral for arsenal DDC as an important positive result. And we've asked countless infectious disease docs treating HIV/AIDS patients that if we had revealed these data of 39, not biomarker events, these are major events, 39 patients having major symptomatic AIDS progression events or death compared to 19 on standard DDI, would this trial have ever been completed? Uniformly, they argued that there would have been prejudgment, that these results never would have been completed. Second example, value trial. The value trial was an important study. Paul Gallo, thank you so much. We had a fantastic independent statistician assisting us on the monitoring committee for this. The value trial was a study being done in hypertensive patients at high cardiovascular risk. And its goal was to compare in this antihypertensive setting two classes of agents, and specifically a member of each class. So within the angiotensin receptor blocker, we had the agent valsartan being compared to a calcium channel blocker, which was amlodipine. And the goal of the study was quite comprehensive in understanding what the effects would be on major morbidity and mortality events. And it was, in fact, to achieve that, a study that required 15,000 people and a 5.5-year duration trial in order to be able to obtain reliable results. So you're with me on the data monitoring committee. Our monitoring committee met regularly, including in August of 2000, where we were a little more than 2 years into the trial of this 5.5-year trial. It was already essentially fully enrolled. It was fully enrolled at this analysis in August of 2000. And at that point, we had a substantial amount of data on MACE events, major macular vascular complications, deaths, myocardial infarctions, strokes. Collectively, we had 700 events. And those 700 events suggested that death rate was 25% higher on the angiotensin receptor blocker compared to the calcium channel blocker. Deaths were 25% higher, MIs were 33% higher, strokes were 33% higher. I love my cardiology colleagues. It was a broad data monitoring committee with multiple cardiologists on that committee, very experienced in cardiovascular research. And for reasons that are based on a great deal of experience, cardiologists tend to be scientific skeptics. You have to show them, show me. They aren't willing to be persuaded, to their credit, by limited amounts of information, even 700 events information. And the committee believed that while these data were very suggestive, they weren't adequately reliable. The study should continue. It did continue. It was, in fact, successfully completed 5.5 years' time. By its successful completion, while at the interim analysis, we had 700 MACE events. By the time we were done, we had more than 4 times that. We had almost 3,000 deaths, MIs, and strokes. And the results were, the cardiologists were right. The final results were very different. That excess death rate on valsartan compared to amlodipine essentially disappeared. And while there was still some excess of MIs and strokes, the relative rates were cut in half. The excess was cut in half. And by the way, we now had deaths on heart failure hospitalization and diabetes, where valsartan was in fact more favorable. So it was very clear that a release of these results in May of '98 would have been very misleading for what the final results were. Let me give one more example before pausing here. This example is of the LIGHT trial. And the LIGHT trial was a study being done for investigation of the combination of naltrexone and bupropion, which has been called Contrave. And Contrave is a weight reduction agent that actually had been proven to have a 5% reduction in weight when it was used in a placebo-controlled trial of patients that were overweight or obese. So a positive result for Contrave as a weight reduction agent. But the study also showed that Contrave had an off-target effect of an increase in systolic blood pressure. And if there's any biomarker in cardiology that is truly quite credible for its prediction of morbidity and mortality, it's blood pressure. And as a result, FDA believed that Contrave did provide weight reduction benefit, but believed that prior to marketing its approval, prior to granting its approval, its effects, its unintended off-target effects on potential cardiovascular risks had to be assessed in a cardiovascular safety trial conducted in overweight, obese subjects that had, in particular, cardiovascular risk factors. And they elected to proceed in a way to facilitate getting Contrave on the market sooner. They proceeded to use what in 2008 had been a paradigm developed in type 2 diabetes, which is to say, eventually you have to rule out an adequately tight margin. In this case, it was set at 1.4, which is a little bit more lenient than in type 2 diabetes because the rates of MACE events are somewhat less in obesity patients. So it was argued that FDA would ultimately want to know that if Contrave is used in these patients, that we could rule out that Contrave could have as much as a 40% relative increase in macrovascular complications and cardiovascular death, stroke, and MI. But that would take 378 events. And so the paradigm that is in type 2 diabetes is we'll let you into the market at a quarter of those events, but you at least at that point have to rule out a margin of 2.0. And so that was the approach. And so the challenge here with FDA's acknowledging and agreeing that you got to keep interim data confidential is how do we in fact allow for the release of information in the first 90 events for a trial that still is only one quarter towards the way of getting 378 events to allow regulatory action without prejudgment. And so this was discussed at, of all the scientific meetings I've ever been to, this was the biggest love fest I've ever been at. It was held in August of 2014. FDA held a Part 15 open public hearing. And FDA, academic community, and industry were all very much on board with the idea of proceeding in this fashion, that yes, in settings like this, we need to have adequate, reliable evidence about macrovascular complications, but we need creative approaches to allow products to get on the market sooner. And so there was great support for the idea. And to do this, support for the concept of having a data access plan, where the data access plan basically said, you're going to create a firewall in the sponsor. The folks on one side of that firewall in the sponsor will be the regulatory reporting group, and they will get access to these emerging data that need to be reported to FDA for filing. The people on the other side of the firewall will be people in the sponsor who are continuing the conduct of the trial, including the management, and they will be completely blinded. Another challenge that we had was how do we in fact allow FDA, in its interest of transparency, to be a player in this process, which they obviously have to be. And so we had high-level meetings. I remember with Steve Nissen, Dave Demetz, I and others went to meet with high-level authorities at FDA, Bob Temple, Alyssa Lavange, John Jenkins, Janet Woodcock, and others, basically to say, FDA, yes, you have a commitment to transparency, but you also believe that in general, data monitoring committees, even with our commitment to transparency, keep interim data confidential when breaches in confidentiality would in fact meaningfully compromise scientific integrity. So FDA, you must agree that your commitment to transparency needs to follow that same principle. And FDA, after some time, agreed, and strongly agreed, in fact, and agreed that in their summary basis of approval in a setting like this, if you had submitted to FDA these data that you would have after 90 events, that FDA would not release the actual point estimate, but FDA would simply report that the data did in fact meet the requirement of ruling out the 2.0 margin, which we argued, Nissen and Demetz and Fleming argued, wasn't material evidence. That could be released because that just means the estimated relative risk isn't more than 1.4, and it has to be less than 1.1 at the end to be a positive result. So that's how we proceeded. And by the way, I'm going to give you an example where it didn't work. where there was a breach in confidentiality, but in every other setting that I'm aware of, it has worked. Industry and regulatory authorities have done a superb job of carrying out this process to achieve the objectives of enabling earlier marketing approval if early results are adequately persuasive, while maintaining confidentiality in a manner to allow the trials to be successfully completed. So what happened in the light trial? I was the chair of the monitoring committee. The first quadrant of data that we needed to get, the data that would be this registrational information, was about 90 events, as you can see, on cardiovascular deaths and stroke and MIs. There were a few extra non-cardiovascular deaths on Contrave, but you put those in too, so you have overall death, stroke, MI. It's still looking very favorable. In fact, not only neutral, but a 36% relative reduction on Contrave. So clearly, this met the criteria of ruling out a doubling. And so following the data access plan and the DMC charter, our data monitoring committee recommended that the data that we released to the regulatory filing group at that meeting could then be released to FDA. And so the process carried on. Interestingly, the sponsor did so, and during 2014, in the next calendar year, in 2014, FDA did grant marketing approval to Contrave. But I'll mention they actually delayed that by several months because they were worried when they became aware that the sponsor in their firewalled away group was much more inclusive than FDA had anticipated would be or should be happening. Nevertheless, they did approve in late 2014. It took 16 months for the second quadrant to occur. So we were in November of 2013, we had the first quadrant. If we look at just the events in the next 16 months, the second quadrant events, the second group of 90 events, wow, this second quadrant looks very different than the first quadrant. In the first quadrant, Contrave had 22 fewer major morbidity and mortality events, deaths, strokes, and MIs. In the second quadrant, it had 17 excess compared to placebo. Okay. Well, when we met on the monitoring committee on March 3rd of 2015, we said, wow, this is interesting. Nevertheless, it's totality of the data here. Our data at the end of the day have to rule out a 1.4. They have to be a point estimate better than 1.14. And this point estimate now is relatively neutral. So it's fine, it's fine. The results are still very consistent with a potential positive result at the end. And so in spite of this unfavorable result in the second quadrant, we recommended trial continuation. But one of the things that we said to each other during that meeting, in that confidential meeting, was, thank goodness for this approach of keeping data confidential with this data access plan, because we now knew that the totality of the data were very consistent with what would have been seen if you had released the data from that first quadrant. Well, by the way, our thank goodness lasted about 3 hours, because 3 hours after our meeting finished on March 3rd, our data monitoring committee was informed that the sponsor had breached their commitment of confidentiality and had released the first quadrant data in a patent filing. And the internet went wild. The internet went wild. We finally have an anti-obesity agent that is meaningfully affecting macrovascular complications. Well, as you could imagine, the data monitoring committee was stunned. The steering committee was stunned, and I expect the FDA was stunned as well. We were informed by the steering committee within 2 to 3 days after that they terminated the trial. They believed that with this kind of information released, that there was no way to carry this trial on with integrity if people that had to be on the placebo arm, and now Contrave was available, would be crossing into Contrave. How would that data be useful then in determining whether you could rule out adverse effects of Contrave? But they and we on the monitoring committee said, nevertheless, even though that decision had been made by the steering committee in March of 2015 to stop this trial, please, when you do, close out the sites, get all the data, and publish the result. And they did. And they published it exactly 1 year later. I encourage you to review this JAMA article and the editorial that followed it. It's incredibly informative. And it shows what those final data were in light. And those final data ended up being about 2/3 the number of events that were intended to be achieved in light when it was finished. But look at these final data in JAMA, the final results, and look how strikingly inconsistent these results are that had led to this remarkable internet wild response that we finally had an effective agent for weight reduction that would reduce macrovascular complications. There is clearly a potential unreliability of interim data. Breaches in confidentiality can definitely lead to dissemination of very misleading results. And by the way, as in this case, can lead to sufficient bias in the conduct of a trial that it can destroy the study, as the light trial was destroyed. The FDA eventually required then a new cardiovascular safety trial be done if Contrave was going to be, in fact, continued in marketing. And I love this quote from Artemis Ward. It isn't so much the things we don't know that get us in trouble, the things that we know that aren't so. And that's one of the really critical issues here with maintaining confidentiality, avoiding prejudgment, because we're worse off if we have an impression of something that in fact could readily be misleading than we would be if we just realized that we don't know what truth is. So the third issue raised by the surgeons, the Australian surgeons, was that the release of data from a concurrent companion trial could render other trials non-influential. And I get it. I mean, I get that this is one of the real concerns that the Australian surgeons experienced. New Zealand and Australia provided a very significant insight in this issue about laparoscopic assisted colectomy versus open colectomy. And yet, a similar study was ongoing in the US by Mayo Clinic, and a similar study was ongoing in the UK, where the US trial and the UK to Europe trial had so much broader populations to draw upon, such that the Australian surgeons were predicting that these other studies were going to come out with their results well before they could finish their trial. And at least if they had access to these data on these short-term effects of whether or not laparoscopic assisted colectomy reduced your days in the hospital, your pain, or your cost, that would be at least something that they could contribute. So it's a question. Is that a valid concern that the Australian and New Zealand surgeons wouldn't in fact have a result that would be viewed as relevant if Mayo and if UK finished first? So interestingly, relevant to this is, again, going back to our HIV experience with NIAID-sponsored trials. Our monitoring committee was monitoring the CPCRA 023 trial, which was being done from 1993 to 95. And we were assessing oral ganciclovir. And the intention of oral ganciclovir was potentially to reduce the risk of CMV retinitis, or symptomatic retinitis, loss of vision, that would be induced by cytomegalovirus. And we were in this 2-year trial, right in the middle of that 2-year trial. We were halfway through. It was July of 1994. And as we met as a monitoring committee to review the CPCRA trial in July of 1994, we were presented with final results from a companion trial that was actually conducted by the sponsor. The sponsor for oral ganciclovir in industry was Syntex. They had done their own trial while we were doing a government-sponsored parallel trial. And they were ahead of us in time. And their results, their final results, were reported out in July of 94. And they looked really encouraging that treatment with oral ganciclovir was having the rate of CMV retinitis. And in fact, surprisingly, maybe even reducing the death rate. So what do we do? What do we do with our trial? And is our study now rendered irrelevant because another study was completed first? Well, we looked at our data, and admittedly, if we had seen the same thing that Syntex was seeing, we might have terminated our trial, but we didn't. Our results were very inconsistent. We had minimal effects in our study on CMV retinitis. And in fact, if anything, mortality was in the wrong direction, but not reliably. So our belief was that this independent study needed to be continued. Furthermore, as we explored these data, we looked at the placebo arm. The rate of CMV retinitis in the placebo arm was about 30% in their trial, 6% in ours. Yeah, well, they had longer follow-up. But when we factored that in, we figured our longer follow-up might increase that 6% rate to 12, but not to 30. And then we discovered that in the Syntex trial, there were ophthalmologists who were doing monthly fundoscopic examinations, and that our fear was what was driving this result could readily be the discovery of asymptomatic retinitis. And while that might have some relevance, we were clinical endpoint people, I think for good reason, and believed at the end of the day that we wanted to know whether oral ganciclovir was having a tangible benefit to patients, was actually affecting CMV retinitis symptoms that were tangible. And so we feared that this might be driven by asymptomatic events. So we carried on. The final results were obtained a year later. And in fact, even less so, there was any benefit. And so our results at the end for our government-sponsored study were very inconsistent with the Syntex trial. And that was in 1994 and 1995. 4 years later, in 1998, I was on an industry-sponsored data monitoring committee. And this was in the setting of multiple sclerosis in secondary progressive MS patients where Berlex was studying beta-seron. And Berlex had done two, in this case, identically designed trials. So unlike that example there that we just had in retinitis, these were two identically designed trials, one done in Europe and one done in North America. And each of them was designed to determine whether beta-seron in secondary progressive MS would reduce the risk of the clinical endpoint of EDSS progression, where in MS, if you progress to 6.5, that's walking with a cane. If you're progressing to 7, that's being wheelchair bound. So it's an important progressive event. And we met right in the middle of our 4-year trial. We were meeting in October of 1998. The European trial that was identically designed to ours was 2 years ahead of us in time. So while our trial was from 96 to 2000, the European trial was from 94 to 2008. So when we met in October of 2008, the European study was actually showing their final results. And the final results of the European study showed a 1/3 reduction in the rate of confirmed progression on EDSS when you're using beta-seron here, versus placebo. So we met. And this was publicized. We knew this result. The public knew this result. The FDA knew this result. And as we met in October, we carefully looked at our data. And our data in the North American setting, which was the only difference between the two trials, is that we were 2 years later, we were in North America, they were in Europe, showed no effect. And we had the benefit also that the chair of the data monitoring committee for the European trial, wasn't me, but the chair of that trial, that committee, was also the chair of our DMC. And so we had full insight about the European results, and we believed that we needed independent replication and that our results were not in any way supportive of the European results. So we recommended continuation. And this was the example that was referred to by Susan or Janet when they were talking about the experience that we had in interactions with FDA. And I had mentioned yesterday, I think it was yesterday, had mentioned that the experience that I had in interacting with FDA when we had ongoing data occurred in this trial because Jay Siegel at FDA had been provided an application for approval by the sponsor based on the European trial, which was logical for Berlex to do. The only data they knew was the European data, and it was quite favorable, quite statistically favorable. So they applied to Jay Siegel and Sieber at FDA for an approval. And Jay, pretty astute fellow, said, well, wait a minute, there's a North American trial ongoing. which is even more relevant to FDA's purview. And he knew that we knew the results of the European study and nevertheless recommended continuation. And so he did seek permission from the sponsor to discuss results with us. We never discussed the actual results, but we were in fact able to confirm his suspicions that our results in fact were going to be importantly informative relative to what the European study had already shown. So we did continue our trial. It was finished in 2000, and the final results were absolutely inconsistent with the European trial results. Siegel and company at CBER, upon access to these data, didn't approve beta-seron, given the inconsistency of results, in particular the negative result or the neutral result in North America. The sponsor, given that these were identically designed trials, when all the results were in hand, did spend an extensive amount of time trying to understand what caused this difference. And it was evasive. It's been very difficult in many settings when you have trials that apparently are the same, yielding different results. It's often not easy to determine. A counterexample to that was the acute coronary syndrome MACE trial of Plato, where there were inconsistent results of ticagrelor and clopidogrel in the US and Europe, where we eventually figured out, we think, that it was based on the interactive effect of aspirin dose, but that's the rare exception. In this setting, the only thing that we ever discovered was when you divided the European data into the first half of the enrolled people and the second half of the enrolled people, the second half of the enrolled people had the identical result of the North American trial of no difference. So the Australian surgeons, in good faith, wished to have access to data. Fortunately, not just the Data Monitoring Committee, but our sponsor, the New Zealand government, the Health Research Council, and the New Zealand surgeons argued that that would be, in fact, compromising, leading to potential prejudgments and compromising the integrity of the studies. The concept that early release would aid enthusiasm is surely countered by the experience at the North Central Cancer Treatment Group in SWOG. The concept that early release will be more timely to reliable results is surely contradicted by the DDI, DDC results in HIV, and by the value trial results, and by the light trial results, where in all three trials, radical differences were seen later in time versus earlier in time. And the concept that if you're not the first one done, then your trial doesn't matter is wrong. Independent scientific confirmation is critical. And ultimately, oral ganciclovir in CMV retinitis and beta-seron in MS, both were not approved, hugely based on the evidence that we obtained by completing trials that had been already underway, that were well underway when other studies were reported prior to them. There is a difference of perspectives, obviously, on any issue as important as maintaining confidentiality of emerging data. Some authors, led by Lilford, argued that why should data arising in a trial be kept secret? A system that perpetuates ignorance violates the injunction that people should not be used as a mere ends to a mean. In our publication of these issues of confidentiality, our New Zealand Data Monitoring Committee wrote an article that basically said this opinion by Lilford and all doesn't recognize that clinical trials must be conducted to address collective and individual ethics. We argued that collective ethics requires ensuring that trials are able to reliably answer the questions they're designed to address, and that even individual ethics require this, because many people join clinical trials for altruistic reasons. And if I had an extra half hour, I'd love to go into some other issues, including Janet Wittes's insight when I mentioned on a previous time when we were working together that patients enter trials for altruistic reasons. She had mentioned the Women's Health Initiative, and when in that trial, partway through, we had the surprising result that you had an actual increase rather than decrease effect with hormones on cardiovascular events, and people had to be reconsented. These women strongly indicated that they were, in fact, in this trial for their daughters and granddaughters for altruistic reasons. patients do join for their benefits, but also join for altruistic reasons. And as a result, we need to protect the ability of the trial to get reliable answers to what they're altruistically contributing to. And I think we had a kindred spirit in the referee for this article. The referee said, in supporting our arguments, that many people don't know the principle or don't think about the second principle of clinical equipoise, where the first principle of clinical equipoise, as quite widely we know, is that it's not proper to randomize unless there's substantial uncertainty in the collective clinical community about the relative merits of the interventions you're randomizing to. And I agree with Richard Pito, it's better to describe that concept by laying it out rather than just calling it clinical equipoise. But the second principle is that it is unethical to alter the ability of a trial to disturb equipoise. And that's what you're doing when you breach confidentiality and lead to prejudgment. And so Damocles in their publication said there's near unanimity that interim data and deliberations of the DMC should be absolutely confidential, and that breaches of confidentiality should be treated extremely seriously. And there are wide statements of concordance by regulatory authorities from EM and FDA, and by academics in NIH and WHO. And this article, again, published by the New Zealand Data Monitoring Committee, addresses these issues in depth. I'm going to give one more example about the breadth of agreement on this issue, and it's a setting of a government-sponsored trial sponsored by the Canadian Institute of Health in peripheral artery disease, where the question was, if you add warfarin to aspirin, do you do better than warfarin than aspirin alone? And as can happen in trials, they can take longer than you think. They can cost more than you think. And the steering committee for that trial approached the Canadian government as that study was progressing on to say, we need additional funding. We're hoping you will add to the funding budget here to allow us to successfully complete this important trial. The Canadian government responded to the steering committee and said, okay, but under the condition that you give us the conditional power results. So conditionally given the current results, what's the probability this will be a positive result? The steering committee and the data monitoring committee objected to sharing the conditional power result with the Canadian government. That would be a breach of confidentiality. The Canadian government responded and said, well, then we're not pleased to go ahead. And the steering committee, which is a great group, it had Sonia Anand, it had Janet Wittes, it had Salim Youssef on it, said, okay, then we're going to get added evidence to support the arguments by the steering committee and the data monitoring committee. So they sent out a survey to 28 experienced clinical trialists internationally from Europe, US, Australia, and Canada, asking the question, do you think that a large randomized trial in which the independent DMC made up of reputable clinical trialists and biostatisticians who carefully monitor the trial, that interim data such as conditional power should be given to the sponsor when requested? 28 said no. There is widespread recognition of the importance of maintaining confidentiality to avoid prejudgment. Having said that,
can I say something?
You bet, Janet, please do.
But of course, we chose who we censor. This was not a random sample of clinical trials.
Fair, but it was a fairly widespread collection.
It was widespread and it was
28.
unanimous.
So, indeed, there are issues that are challenging issues. We've talked about one, which is these cardiovascular safety trials. How do you release interim data for regulatory approval and finish the study? A similar setting is in accelerated approval, where from an FDA perspective, full approval is based on a clinical endpoint like overall survival, but you can get accelerated approval by having effects on a proper biomarker, making it reasonably likely to predict benefit, like progression-free survival. So let's say you're doing a trial that is powered to achieve 300 PFS events, but then to go on and eventually obtain 400 survival events, where if the biomarker results are positive, you get marketing approval, conditional approval, accelerated approval, and then ultimately the validation occurs when the survival results reach the 400 events. So suppose you do this accelerated approval strategy and you use a single trial, and it's a very good strategy to do it, to get the PFS results. And when you have those 300 PFS events, suppose you have 150 deaths of the 400 deaths. How do you proceed if the PFS results are really favorable if you want to maintain the integrity of the survival data? Well, one obvious thing to say is when you have your 300 PFS results, do an interim analysis of survival. And if that's positive, that's great. You can file the FDA not just for accelerated approval, but for full approval. Yes. Well, what if when you do that interim analysis, you have a positive result on the 300 events for PFS, but the 150 survival events don't cross the boundary? We argued in an article with Susan and Dave, recently published. It's on the course website, DMC current issues, that in such settings, the information on the clinical endpoint need to be kept confidential. So release the information for regulatory approval on PFS, but maintain confidentiality on survival data, as you always should, as the registrational endpoint until for full approval, until you have persuasive evidence. Another issue, and that is a complicated issue: can the sponsor have access to pooled data? Some of us have argued that routine widespread access would be inappropriate. So a data monitoring committee open report that shares information broadly with the sponsor and others that would have access to the open report should not have pooled data on efficacy and safety measures, but that the pooled data should be available on a need-to-know basis. So medical monitors will have access to data during the course of the trial to assess SUSARs or SAEs. Caregivers, obviously in unblinded trials, will know what the intervention arm is and what the results are for patients at their clinic, but should only know what the results are for patients at their clinic. Pool data should be available to somebody on a study team in an event-driven trial, because it's not the sample size that's fixed, it's the number of events that's fixed. So if it's a survival trial, I need 400 events. I think I have 600 patients. If the event rate's less than I think, I have to adjust the sample size to restore the ability to get my 400 events in a timely way. Absolutely right. And that's a need to know. But those exchanges should be occurring in a manner that is restrictive to allowing those purposes to occur. So there was an example of a trial that was an open label trial, and an investor got the names of, I think, 15 of the clinics that were involved, called them, and found out what was happening in each of the 15 clinics and put that together. That's a breach of confidentiality. I blame the sponsor in that case for not ensuring that the caregivers knew that their knowledge, by the nature of the unblinded trial, to the emerging data was purely for purposes of managing those patients and couldn't be shared with other caregivers and others beyond the medical monitor and the data monitoring committee. And access to pooled data. If we have a pancreas cancer trial, we're trying to improve 2-year survival, and you expect 10% 2-year survival in the control arm. If you show me the pooled survival is 5%, that's pretty informative that this is not working. Or you show me the pooled survival is 20%, that's pretty informative that it is, or at least I might be interpreting that. Recently, I got a phone call from a sponsor who wanted to engage me as a consultant and they let me know that they were conducting an ongoing trial with a monitoring committee that was keeping data confidential, but they had access to pooled data. And they took the pooled data on the primary endpoints and the secondary endpoints, and they went out and found extensive external registries that gave them a good sense of what should happen in natural history. And they concluded that their pooled data in the trial didn't look very different from the registry data for the primary endpoint, but looked really different for some secondaries. So they wanted to confirm that, from my perspective, that it was okay for them to proceed in that way and change the primary endpoint. And as you might imagine, I went ballistic. It absolutely is not okay. Why in the world do they have access to that pooled data? That is, in fact, potentially somewhat indirectly informative about efficacy. And as a result, you absolutely, once you have indirect access, if not direct access, are not in a position where it's proper to use that information to change the trial design. So open reports, we've talked about this briefly in the first 2 days of the workshop, are really important. They're provided to study teams. They include pooled data across treatment arms on quality of study conduct measures. How are we enrolling? What's our adherence? What's our retention? And what's our event, but not specifically information that's pooled on efficacy. So not sharing pooled data on the primary endpoint or pooled data on secondary endpoints or on safety. The closed report would provide all that information broken out by intervention group, as well as efficacy and safety data. And an issue that great to hear from Susan and Janet in the last couple of days, where the DMC needs to be unblinded, and time is short here, so I'll go quickly through this. In our experience, again, on this Division of AIDS Data Monitoring Committee, on which I served for 20 years, monitoring 50 trials, we evolved in our thinking. We were pioneering in many ways, working with NIAID and Susan and the Data Monitoring Committee on procedures for how best to achieve and to preserve the integrity of proper monitoring of trials. In early days, people realized, as was stated by Janet and Susan, that monitoring committee is smart. If they're looking at a product, give them an A/B coding and they'll figure out which is the active and which is the control. So we'll see more safety issues on Arm A. It's obvious that it's in the domain of what we expect this to happen with this intervention of soft target effects. Arm A is clearly the experimental. And by the way, you're almost certainly correct. If in fact efficacy was coded AB, you would know everything. So in the early days, to prevent us from knowing everything, safety was coded AB, efficacy was coded XY. Okay, well, there are more safety issues in Arm A, but efficacy is better on X. We presume that A and X are the same arm, so we continue on. But wait a minute, maybe we're wrong. Maybe the active arm is A and the active arm is Y, and so we have very unfavorable benefit to risk. And we objected, and Division of AIDS agreed with that objection, and since 1988, the Data Monitoring Committee has been unblinded. And that unblinding has certainly been critical to us in understanding benefit to risk. It's also been really critical to us in understanding trial integrity issues. Just very quickly, I was on an international, a data monitoring committee for an international trial. And we had gathered together back in pre-COVID days. We had an in-person meeting. And not only was our data monitoring committee completely assembled, outside in another room, the steering committee was completely assembled for a meeting about this trial. And we monitored the results. Our intention, as was standard, was that when we finished our closed session, we would go out to the open session, report our recommendations to the steering committee. Well, what we saw in that trial, in that major international trial, were 50 excess deaths on the experimental arm. And while that didn't prove harm, it clearly ruled out benefit. And so we were prepared to recommend termination of the trial to the steering committee. Yet as we explored the data in greater depth, we were troubled by the fact that there were a larger number of safety issues in the control arm, where we surely would have expected that the experimental arm with its mechanism, we wouldn't have expected it would have had 50 excess deaths, but we would have expected that it should have had excess, not a reduction in some of these safety issues. So we said to the independent statistician, could the code be backwards? Is it possible that what you're telling us here is the active arm is really the control arm? And the independent statistician said, I don't think so, but it's a fair question. We will look into it. We said, please pull several patients and check the vials as to what they actually received and make sure that's what your report is telling us is their treatment arm. Fine, that was the agreement. So we left the closed session. We went into the open session. We met with the steering committee. We had discussions about quality of trial conduct, but we said, please grant us a few extra days to formally give us the recommendation about trial continuation. They said fine. And so we moved forward. 4 days later, I'm sitting at my office at UW and I get a phone call from the independent statistician saying, Fleming, you guys were right. The code was backwards. And so I'm doing backflips in my office that we had prevented the inappropriate release of those data. But basically what we did is we just got on the phone with the sponsor and said, continue on. That was it. Because while there were, in fact, now not 50 excess deaths, 50 fewer deaths, that wasn't sufficient to establish definitively efficacy, but it surely was appropriate to continue. So a fully informed data monitoring committee is really critical. There are settings where the codes have been backwards. In the 200 DMCs I've been on, it's happened, I think, 4 times where the codes were incorrect, either backwards or were nonsensical, as I was referring to yesterday in the discussion. Fortunately, I can say I don't think any of those instances have occurred in the last 7 to 10 years. I think it was all previous to that. But another reason for the data monitoring committee being unblinded is nicely shown by the cardiac arrhythmia suppression trial. And in this CAST trial, we were studying econide and flecainide against placebo. And as many of you know, when you have an MI and you have an arrhythmia, that's a bad thing. That's a significant risk factor for torsade and sudden death. And in those settings, there was great interest in suppressing arrhythmias with the idea that obviously that would greatly reduce these risks of sudden death and torsade. Yet, while this was sufficient evidence, it wasn't FDA approved, but there were hundreds of thousands of people in the US alone who were using econide and flecainide on this basis. It was recognized by many that this wasn't a reliably established beneficial approach. And so the cardiac arrhythmia suppression trial was conducted in several thousand people, randomizing them to the antiarrhythmics, econide flecainide against placebo. Interestingly, in this setting, the monitoring committee was blinded to the actual treatment arms. They were provided information by A, B only, and they weren't told what A and B were. So at an interim analysis, they were informed that there were 19 sudden deaths on one arm and 3 on the other. And I think the committee, I'm assuming, I wasn't part of this process, but I'm assuming they believed that it's pretty apparent that the 19 must be placebo and the 3 must be the antiarrhythmic, that they said, go on. Interestingly, while the monitoring committee was blinded, the statistician that was the lead study team statistician was unblinded. And when the 19 versus 3 became 33 versus 9 sudden deaths and 56 versus 22 overall deaths, the study team statistician said, I think the DMC probably should know this, and called an emergency meeting, informed the data monitoring committee, that then immediately terminated the trial. And it will forever be unknown what the monitoring committee would have done earlier in time, but it surely can be argued that their ability to appropriately safeguard patient interests was compromised in this setting by virtue of the fact that they were only provided information by arms A versus arm B. So the DMC having access to unblinded data is certainly critical to being able to make more timely recognition of emerging patterns, as in the CAST trial, and in understanding safety in the context of benefit to risk, as we've been mentioning. As I mentioned, that antiplatelet agent that might induce major bleeding, that's a safety risk. And yet, if you have a significant decrease in ischemic events or even mortality, that's positive benefit to risk. And it facilitates their ability to detect irregularities, as was important that we were unblinded in that international trial. So to sum up on confidentiality issues, it is critical to trial integrity to maintain confidentiality to reduce prejudgments. The DMC, though, should be unblinded. Another issue that I'll just briefly mention, there are rare settings where the data monitoring committee may believe that the sponsor's dissemination or lack of dissemination leads to serious ethical and scientific concerns. What can you do in that case? We discussed one of these in the second edition of Ellenberg, Fleming, and the Betts. It's in chapter 11 of that, where a sponsor did post hoc subgroup analyses without providing a proper dissemination of the context for those results, claiming benefit from those subgroup analyses when the totality of the data and subsequent confirmatory evidence showed that that was completely spurious. How does the DMC proceed? There's settings that I'm aware of where a data monitoring committee on 2 occasions has recommended termination of the trial for excess deaths, and the sponsor, with their discretion to not do so, didn't. How does a monitoring committee proceed? And we could argue that it shouldn't be the monitoring committee's purview to simply breach their commitment to confidentiality in those settings and release data. And yet they have an ethical imperative to protect the public. And so our belief is that in those rare settings, there should be a mediation process. That in these rare settings, if the DMC believes that there are serious ethical concerns with how the sponsor has disseminated or not disseminated information, that independent mediation process could be of assistance. So let me pause. Any further thoughts? In fact, I think I'm going to jump ahead now that I look at the clock here. I realize here we've got about 15, 20 minutes left. I want to quickly touch on a couple of other issues. And among these are issues that also are key to DMC independence. And one of them is the meeting format. So I've mentioned this Division of AIDS, NIAID Data Monitoring Committee that's monitored over decades from the 1980s on. And that group was part of the process that said, let's modify the meeting format to not just have closed sessions, but to have open sessions, because investigators and sponsors and regulators have so much to contribute. So even while they have conflicts of interest, we need their insights. And so that group at Division of AIDS evolved a process of having a closed session, followed by an open session, followed by a closed session, as a way to maximize the insights while maintaining confidentiality. And there's no single right way to do this. Many start with the open session and have a closed session. Many of us have argued, though, that we shouldn't get rid of that initial closed session. It's, in my view, very important to helping to ensure that the chair realizes they are in the lead role here, not others that are running the open session. And furthermore, you can use that closed session to be better prepared for how best to proceed in the open session. A simple example of this was our Division of AIDS Data Monitoring Committee that was monitoring a study looking at an experimental use of fluconazole to reduce the risk of serious fungal infections. And the study was designed for a 4-fold reduction. They were powering for a big effect, which meant they only needed 25 events. And we were 2/3 of the way through. We had 16 events. And they were 14 of them were on the control arm and only 2 on fluconazole. So fluconazole didn't just have a 4-fold, it had 7-fold reduction. And it actually crossed, just barely, but across the O'Brien-Fleming boundary for benefit. So, you know, we would think that we should terminate early. And this comes back to the examples that Janet and Susan were talking about earlier as well. There clearly is a great deal of influence by a monitoring boundary, but it's not a rule. And in this particular setting, while at this 2/3 of the way, 2/3 information interim analysis, there were 12 fewer serious fungal infections on fluconazole. There were 14 excess deaths, 45 against 31. Our monitoring committee said, well, gee, serious fungal infections matter, but deaths count more. And so we were uncomfortable terminating the trial. So we went to the open session, told this to the sponsor. They agreed that we should continue, so we did. Well, you should be jumping up and down right now saying, Fleming, you can't tell the sponsor. And you're right, we can't. So to tell you the whole story, what we did in the first closed session is we formulated many things to talk to the sponsor about in the open session, including this issue, where we came up with 6 scenarios that we ran by the sponsor asking what they would do. And one of them, scenario 5, was fairly close to the actual results. So we went to the open session, we went through that process, they told us what to do, we said thank you, they went home, we went back to our closed session. They had said continue on for that, we agreed, we continued on. When the study was done a year or 2 later, we met with them. We said, you know, 2 years ago when we said this to you, do you have any idea what we were doing? They said they had no idea. They had no idea what we were talking about. We were able to use the closed session in a way to skillfully position ourselves to learn as much as possible in the open session from people that had complementary expertise without unblinding them. So another critical issue, the charter, it needs to be properly formulated. And as Janet and Susan have pointed out, the charter is really a key document. It tells the primary responsibilities of the DMC, the membership of the committee, the timing and purpose of the meetings, procedures to maintain confidentiality, the open and closed sessions, open and closed reports. The open sessions have open reports, the closed sessions have the closed reports, and then open and closed minutes, the recommendations, the monitoring boundaries. All of these are laid out in the DMC charter. And the sponsor, but very much also the DMC, has shared responsibility in finalizing that charter. So what are the issues here that our working group on developing best practices felt were important to address here for the charter? And the key issue that is concerning to us, as I think Dave Demetz would say if he were here, is the dismay about how charters are becoming longer and longer documents. Instead of being a 15 to 20 page document, they're often 35-page documents. They're written in a very legalistic manner, setting up rigid requirements, limiting the number of looks, indicating that the DMC can only look at safety data to avoid spending alpha, indicating how we're going to vote and the voting process and what will happen, and indicating that whenever there's any change to this legalistic DMC charter document, we need to send this out to the DMC members and have them not only re-sign the charter, but to do it with wet ink that has to be sent by express mail. I kid you not. I've been on trials where on 1 to 2 occasions per year, I had to do a wet ink signature of to approve a revised charter. And the issue is, it's completely missing the scientific and ethical purpose of the charter. The charter is not a rigid legal document. The charter is intended to articulate principles that provide guidance to the DMC process, and a DMC needs to have the flexibility to deal with the inevitable unexpected challenges. And they have the guidance from that charter, but they then use their judgment. And it's not a rule book. So another aspect of independence is the DMC contracting process. And I'll note that it's certainly very important that real and perceived conflicts of interest be identified and minimized. And Susan has nicely pointed out that when we are selecting data monitoring committee members, it's really important to do so in ways that allow for the independence of the DMC. And so while some of the most informed people we would love to have with us, we can only have them with us in open sessions, because the membership of the DMC should be independent of the sponsor and independent of those who developed the products and who wrote the protocol and who are treating the patients and who are the regulatory authorities that will be evaluating the data at the end in applications for regulatory approval. So those are really important issues. There are other issues too, though, that influence conflicts of interest. Ideally, when we have a meeting, I'd love to meet in Maui. I would love it to meet in Maui. Nowadays, we meet on the internet, but when we met in person, and there are real advantages to meeting in person when we can, the O'Hare Hilton was my most well-known destination. It's convenient. It's not, an exciting place to be, but it gets people together in an efficient way. It's interesting. I know some people have said, gee, Fleming, you're too, hung up on rigors here. When I say I'm not comfortable having a pre-meeting dinner with the sponsor. But I think about this. I was thinking about this. In serving on an FDA advisory committee, and a number of us on this call have been on FDA advisory committees, can you remember when you're serving on an FDA advisory committee, ever the night before the advisory committee having dinner with the sponsor that's presenting to the committee? Or even having dinner with FDA? These aren't social situations. A judge is independent. You don't meet with the judge socially before the judge tries a case. And and I don't want to be overly stringent here, but it's better to recognize that a monitoring committee is an independent entity. They may have a pre-meeting dinner amongst themselves or with the independent statistician, but it doesn't seem proper for that to happen with sponsors. Another issue that's not a huge issue, but it's one of some concern is how do you engage sponsor? How does a sponsor engage DMC members? And it's obvious that you have to have some kind of a consulting agreement. It's concerning to me that very often the consulting agreements that were sent are generic consulting agreements that would be used to engage me as a consultant for the company. So the consultant, DMC, rather a company may call me and say, I want your help in helping design this trial or in helping to interpret the data in the trial. Well, I realize I'm working for the sponsor. And I realize that in those types of agreements that there are issues of indemnification. And on some occasions, people might be expected to indemnify the sponsor. It's a very different circumstance when the sponsor is engaging you as a DMC member. I don't consider myself working for the sponsor. I consider myself working for patients and for caregivers and working on behalf of everyone else for whom we are expected to be trustworthy, including the IRBs and regulatory authorities and investigators and patients. So it is important that this process be done in a way, ideally, to have a more independent scientist agreement, and if possible, but not necessarily, where the people could be engaged by an independent entity, such as the steering committee. So these aren't absolutes, but they are additional aspects that influence the nature of the interaction, and the interaction should be very much one of independence. I would spend time, but after a fantastic discussion by Janet yesterday, there's not a need to spend time about another important issue that was carefully considered by this working group looking at developing best practices, and that's the Independent Statistical Reporting Group, or SSTAC. And Janet has covered this so nicely. I'll just take 30 seconds to point out that the Data Monitoring Committee, as Janet was indicating, is hugely reliant upon the accuracy and timeliness and approach in clarity of the information in DMC open and closed reports that are generated by the independent statistician. It's critical that that independent statistician have a sufficient depth of knowledge and experience with trials to allow us to get timely, reliable, and readily interpretable insights. And if you wanted to know more about what I mean, I'd say review that fantastic presentation by Janet yesterday. So in essence, what we're saying is in best practices, amazing things have been done in the evolution of use of data monitoring committees in clinical trials. And in the last 30 years, industry has joined NIH and government sponsors and been a major player in the development of many of these very important evolutions that have been positive. At the same time, there are concerns that as we expand use, we do so in ways that achieve best practices, that empower data monitoring committees to carry out their mission effectively, which is to safeguard patient interests and to enhance and protect trial integrity, which does mean being able to preserve sole access to emerging data and does mean to preserve their confidentiality, their independence. And so all of these issues are really critical. We need better training. We need proper indemnification. We need confidentiality, although the DMC members should be unblinded. Charters should not be rigid documents. They should be setting up guiding principles. We don't vote. We don't vote. If I'm on a committee with 5 members and 3 say stop and 2 say continue or vice versa, we're not done. This is not a political process where majority rules. We have a much greater responsibility to all of the people that are giving us their trust to proceed in a manner beyond majority till we have consensus. I don't think I've been on a committee in 30 years where we weren't able to achieve that consensus. And I bristle when I see indications of the use of voting or somebody saying we need an uneven number of members so we can vote. It's a consensus development process. The chair's leadership is critical, and beginning with a closed session, I think enhances that. Contracts need to be set up in ways that recognize we're independent scientists. And as we heard so clearly yesterday, the ISRC needs the experience, the access, and the flexibilities. And real quickly at the end, I completely support the fact that regulatory authorities should not be on a monitoring committee when they will have responsibilities when that study's done to determine whether these data are sufficient for marketing approval. But that doesn't mean that regulatory authorities couldn't be on monitoring committees for areas independent of their regulatory responsibility. For example, of a behavioral intervention in an NIH trial. And we rely so much on regulatory authorities for guidances. And we're asking for those guidances to come from people who have not been able to serve on the committees that they're in fact setting up best practices, procedures, and guidances. So some of us have thought creative approaches to enhance regulatory authorities' access will be very important to enhance their insights when they're developing guidances. I will shamelessly say we've covered a whole lot in these 3 sessions, these 3 days. The Data Monitoring Committee second edition is now out. For years, I've been saying people, don't buy it, because for the last 10 years, Susan and Dave and I have been working on the second edition. It's now out. I'm saying do buy it. There's so much that we've covered. There are a number of things, though, that we haven't, and there's much more in there. I mentioned from Katie's perspective, thank you so much to Katie for making all of this possible. She also noted that you should be reminded that there's a post-survey link for Module 9 that's posted to the module webpage and to the Slack channel. And so we would encourage you to gain access to these now the course is done, and any insights you have would be greatly appreciated. I am, and we are all collectively so grateful to Susan and to Janet for their remarkable presentations on Monday and Tuesday. And as I've already mentioned, to Katie and Deb Nelson for putting all of this together, and very much to all of you as attendees. I often say to people say to me, Fleming, you know, we're grateful that you did this. And I say, I'm grateful that you're here. You're the future in many cases. We need you, all of you who are attendees. We need you. We need you as Data Monitoring Committee members. We need you as ISRG members. We need you as caregivers and IRBs and investigators, and very much as sponsors and regulators, ensuring that data monitoring committees that are in place in many instances where you have influence on those are able to, in fact, carry out best practices. So I would like to end by thanking all of you who participated here today and Monday and Tuesday for what you've done, and very much thank you for what you will be doing in the future, because data monitoring committees have such an important mission, and you will be able to have an important influence on it and enabling them to achieve that. So thank you all.